صباح الخير طلاب. The second our talk this morning is metabolism of glycogen. We're going to explain some biomedical importance for the glycogen and what is the glycogen and what's the chemical formula or this type of carbohydrate with some of disease, and where does it store this form of the polymeric carbohydrate, and the glycogenolysis and the glycogenesis synthesis and the hydrolysis of the glycogen. Let us start with the some introduction, biomedical introduction about the glycogen. Okay. Biomedical importance. Glycogen is a major storage of the carbohydrate in animals as a fast mobilized fuel store. Therefore, this type of the carbohydrate, which is glycogen, is could be considered as a fuel store, but fast fuel store. يعني بسرعة ينطينا الطاقة. Storage of the carbohydrate in the plants as a starch, while storage of the carbohydrate in the animal, storage of the carbohydrate in the animal as a glycogen, But you have to know just the excess amount of the glucose is converted to polymeric forms. Any excess amount of the glucose will be converted to this form, glycogen, where in vertebrate or animals. Therefore, two classes or two ways to store the carbohydrates in the animal as glycogen, but just the excess amount, and in the plants as a starch. This is the important points in this note or page. Okay. Now, where is the glycogen and how much? Glycogen is present in a large amounts in muscle cells, but the percentage 1 to 2% of the weight of the muscle, and in the liver cells 10%, greater than the percentage of the glycogen in the liver, 10%, it is greater than for the muscle. It is very clear why, because although the liver content of glycogen is greater than that of muscle. Why? Because the muscle mass of the body is considerably greater than that of the liver. Therefore, despite of the amount of the glycogen in the muscle is greater than the amount of the glycogen in the liver, but as a percentage, the contents or the liver contents is greater than the muscle contents because of the mass. About 3/4 of the total body glycogen is in the muscle, but as a percentage is less than the liver. Therefore, because of the mass, the percentage will be converted. Now, where it is stored in the cytoplasm in the form of large hydrated granules. Have a look for this diagram. The glycogen will be stored inside the liver as hydrated granules. This is hydrated granules. Okay, large amount of the hydrated granules. Okay, here just we have to know the percentage of the glycogen is in the liver more than in the muscle, despite of the amount of the glycogen in the muscle is more than the amount of the glycogen in the liver. Okay. Now, this amount of glucose, if were dissolved in the cytosol of the hepatocyte, its concentration would be about 0.4 molar, and this enough to dominate the osmotic properties of the cell. What does it mean that? We need about 0.4 molari, 0.4 molari just will be enough to dominate the osmotic properties of the cell. Now, glycogen is very important inside the cell. Why? Because we'll give a level, certain level of the glucose inside the cell, and this certain level, which is about 0.4 molari, enough to dominate the osmotic properties of the cell. And you know this is very important property for the cell, osmotic properties. Okay? So, have a look for this table. This table indicates storage of the carbohydrate in certain or person has 70 kilograms. Now, we need to focus on the 2 points here: liver glycogen and muscle, liver glycogen and muscle glycogen. Liver glycogen as a percentage 5, while muscle glycogen 0.7 is less than the liver, despite of the amount of the glycogen in the muscle 245, while the amount of the glycogen in the liver 90 gram. But because of the mass of the liver less than the mass of the muscles, therefore the percentage will be higher in the liver. Okay, and there's a certain amount of the glycogen in the extracellular fluid. Okay. Now, what's the function of the glycogen? What's the function of the glycogen in the liver? In the liver, 2 functions: store and export. Store and export. Store, that means any excess amount is coming from the GI system and from the digestion will be stored. This carbohydrate or glucose will be stored as a glycogen. Export between meals. Any deficiency in the level of the glucose or blood glucose, the liver will be hydrolyzed the glycogen to form the glucose to maintain the normal level of blood glucose. That means 2 functions, but between meals. Export between meals. It's very important to mention here, we need 12 to 18 hours as a fasting. The liver glycogen is totally depleted. Okay, we need at least or between 12 to 18 hours as a fasting to reduce or all the amount of the glycogen inside the liver. Okay, while muscle glycogen, as I said, is a readily available source of the glucose. Now here, quick source of the energy, while in the liver, liver glycogen could be considered as a slow source of energy, but in the muscle, quick source of energy. Of course, for a lot of processes like aerobic or anaerobic metabolism within the muscle itself. Okay. But another very important points here. Muscle glycogen can be exhausted in less than 1 hour during vigorous activity. Now, you have to know the glycogen in the muscle could be depleted with vigorous activity, but we need 1 hour. But the glycogen in the liver, we need. A person must be fasted 18 hours to reduce or to take all or to use all the glycogen in the liver. Therefore, as a total, we need 19 hours, at least 18 plus 1, 19 hours to make sure the amount of the glycogen inside the human body was depleted. Okay? Therefore, any person need to make regime, يعني اللي يسوي regime, need 19 hours, 18 hours fasting, then 1 hour walking or make any exercise. By this way, we will make sure all the glycogen or carbohydrate has been depleted. Therefore, the human body will make substitution instead of the glucose yes, glucose coming from the digestion or from the hydrolysis of the glycogen, will make a substitution by another compounds like ketone bodies or triglyceride in the adipose tissue. By this way, will reduce the weight of the person. Okay? Here, this figure 2, the main sites of the storage of the glycogen inside the human body. Liver has glycogen and muscle has glycogen. But the glycogen in the muscle could be considered a quick source of energy, transfer to glucose 6-phosphate, then glucose enter the citric acid cycle and or hydrolysis, glycolysis and citric acid cycle to give the energy. Also glycogen here, but this is very slow. Why? Because the glycogen in the liver will be converted to glucose 6-phosphate, and we need the glucose 6-phosphatase as enzyme to convert the glucose 6-phosphate to glucose. This enzyme, the activity of this enzyme depends on the insulin. Therefore, if there is deficiency in the glucose inside the bloodstream, that means we don't have enough amount of the insulin to activate this enzyme to transfer the glucose 6-phosphate to glucose, then enter a cycle to give the energy. Therefore, the process will be slow. This is very important to support or to keep the glucose inside the liver to support another very important organs inside the human body like brain, between meals or starvation or fasting. The brain needs glucose to make energy. Therefore, the hydrolysis of the glycogen to glucose will be slower to support, by this way, to support the amount of the glucose in other organs, especially brain. Okay. Okay. Now we've got some biomedical importance about the glycogen. Glycogen is the polymeric form of the glucose or carbohydrate, or storage form for the carbohydrate, where inside the liver in the vertebrates or animals. Glycogen could be considered a slower source of energy, while inside two sites to store the glycogen, muscle and the liver, but muscle quick source of the source of energy, glycogen in the liver, slower source of the energy. And now let us take one example about the glycogen storage and some of the disease. Glycogen storage disease, mainly group of inherited disorders, of course characterized by the two properties. Okay, maybe because of deficient mobilization of the glycogen, that means movement of the glycogen, there is a defect in the movement of the glycogen. And the second cause, abnormal forms of the glycogen will be deposited. Deposition of the abnormal forms of the glycogen. Two causes, very clear: movement or deposition of the glycogen, where in the liver, leading to sometimes muscular weakness or reach to even death. For example, we've got very famous disease, von Gerek's disease. Von Gerek's disease. This disease will be because of deficiency in the glucose 6-phosphatase. That means because of deficiency of this enzyme where in the liver. We don't have enough amount of this enzyme in the liver. Therefore, this deficiency will lead to form or make the von Gerek's disease. Of course, the symptoms are liver cells and renal tubular cells loaded with the glycogen. Yeah, because we don't have this enzyme, therefore all the glucose 6-phosphate will be still inside the liver. Therefore, one of the clear symptoms for this disease, Gerek's disease, loaded or cell loaded with glycogen. Hypoglycemia, yes, because of we don't have glucose inside the liver to get out to the bloodstream. All the glucose is phosphorylated glucose, is still phosphorylated glucose as glucose 6-phosphate. Lactic acidemia, yeah, the pH of the blood will be decreased, the acid will be high, because of the human body will make substitution instead of the glucose, make substitution for the glucose by the ketone bodies or lactate, or amount of the lactic acid will be increased in the bloodstream. Ketosis, yes, because of three types of the ketone bodies as a substitution for the glucose. For example, beta-hydroxybutyrate, butyric acid, or beta-hydroxybutyrate, and hyperlipidemia, because the human body will make substitution by the adipose tissue. Triglyceride in the adipose tissue will be hydrolyzed to form glyceraldehyde, and then glyceraldehyde will transfer to acetyl-CoA to enter the citric acid cycle to give the energy in addition to CO2 and H2O. This is the main symptoms for this disease because of the deficiency in this enzyme. Okay. What's the main symptoms for this disease? Patient with this disease has huge abdomen. Why? Because massive enlargement of the liver. Massive enlargement of the liver. And hypoglycemia. There is a pronounced hypoglycemia, but between meals. Furthermore, the blood glucose level does not rise on administration of epinephrine and glucagon. Now, patient will with this disease has huge abdomen, يعني تنتفخ بطنه بسبب massive enlargement of the liver. ليش؟ لأنه يصير عندنا loaded with the glycogen فالmass of the liver will be increased راح يزداد حجم الليفر فيصير عندنا huge abdomen in addition to that hypoglycemia between meals. Okay? We're going to explain this point. Furthermore, the blood glucose level does not rise on administration of epinephrine and glucagon. Why? We're going to explain that during the synthesis, during the glycogen synthesis or glycogen phosphorylase or glycogenolysis or glycogenesis, يعني during synthesis and hydrolysis. In the fact, glucagon and epinephrine increase the cyclic AMP, which cyclic AMP is very important in the synthesis and hydrolysis of the glycogen. Of course, cyclic AMP decrease the glycogen synthesis and increase the glycogen hydrolysis. وبالتالي the effect of this hormones epinephrine and glucagon didn't affect or don't affect this two hormones on the level of the glucose. That means glucose level does not rise. Glucose level does not rise. يعني basically these two enzymes increase the hydrolysis to give the glucose, but because of the deficiency of the glucose 6-phosphatase, we don't have the glucose. Therefore, the level of the glucose will not be increased. Okay. The absence of glucose 6-phosphatase, this enzyme, in the liver causes hypoglycemia, as we said. Why? Now let us go inside the liver. Because glucose cannot be formed from the glucose 6-phosphate. We don't have this enzyme, therefore we don't have a glucose, cannot convert glucose 6-phosphate to glucose. This phosphorylated sugar doesn't leave the liver because of its phosphorylated cannot pass across the plasma membrane. The presence excess glucose 6-phosphate, this is triggers an increase in glycolysis in the liver. Therefore, the glycolysis will be increased inside the liver, but this leading to high level of lactate and pyruvate in the blood. Why? Because we don't have enough amount of the glucose. That means we don't have enough amount of the energy to transfer the glucose to CO2 and H2 and energy. Therefore, pyruvate will be converted to lactate. The excess amount of the pyruvate and lactate will be get outside the liver, then increase this level inside the bloodstream. Okay. Now let us some information about the biological structure of the glycogen. Glycogen is a prized polymer, as we said, polymer of a glucose residue. That means the essential unit of the glycogen is glucose residue. But this sequence or series of the carbohydrate, most of the glucose units in glycogen are linked by alpha 1,4 glycosidic bond. Now, the sequence, each one molecule of the carbohydrate is bonded. Have a look for this figure. The unit of the glucose is bonded with another unit of the glucose by 1,4. This position 1 and this position 4. 1,4. But alpha, this carbon alpha or beta, here alpha. Alpha 1,4 glycosidic bond. Glycosidic, that means the bond by the oxygen atom. Therefore, this sequence, straight chain of the carbohydrate, each unit bonded with other by alpha 1,4 glycosidic bond. But each 10 units of this uh, uh, sequence, each 10 units, there is a branch. There is a new branch. But the bond between alpha 1,6 glycosidic bond. Now here, this is still alpha 1, the hydroxyl group alpha, the direction of the hydroxyl group alpha, zinc beta, 1, the carbon number 1, 6 here, here there is carbon number 6 in this unit. That means alpha 1,6 glycosidic bond. This is the structure of the glycogen. There is two straight chain, but the second chain after 10 units of the first chain by the branch bond alpha 1,6 alpha 1,6. Okay. Now let us enter the central point in our subject, the pathway of a glycogen biosynthesis. Pathway of a glycogen biosynthesis. We're going to take just one step and leave the other or all the left in the next talk. Okay. As in glycolysis, we know what does it mean glycolysis. Hydrolysis of the glucose, but must be start with phosphorylation of the glucose by phosphate group. A glucose is phosphorylated to glucose 6-phosphate. A glucose is phosphorylated to a glucose 6-phosphate. Here, to glycolysis and pentose phosphate pathway, as in this process. Catalyzed by hexokinase in muscle and glucokinase in liver. That means synthesis of the glycogen sometimes in the liver and sometimes were in other organs or bloodstream. But we need also, the first step is phosphorylation of the glucose using two types of the enzyme. Inside the liver, glucokinase enzyme will do this step. But in the muscle and other organs, we need hexokinase. That means we got two types of the enzymes, one of them especially in the liver and the other especially in the outside the liver. And as I said, this enzyme is affected by the insulin hormones. Therefore, there is a clear relationship between the level of the glucose in the bloodstream and activity of this enzyme. But this is the explanation. This step it's outside the scope of our subject. This will be explained in clinical biochemistry in stage 5. Here, two enzymes, glucokinase, put the glucose, put the phosphate group in the glucose, but phosphatase release of the phosphate group. That means the action of the kinase is opposite for the phosphatase. Kinase will add the phosphate group. Phosphate will release the phosphate group. This is very important. You have to put this in your mind anytime. In addition, we've got also, have a look for the magnesium plus 2. It is a good coenzyme for the glucokinase. Therefore, this is the first step to start the pathway of a glycogen biosynthesis or glycogen synthesis. To this point, we're going to stop on the next lecture we will start all the steps of the biosynthesis and hydrolysis of the glycogen. Generally, we need to know glycogen as a biomedical field. In the biomedical field, glycogen is very important to store the excess amount of the glucose coming from the GI system will be stored as a glycogen inside the liver. Glycogen is a source for the energy, but we've got two sites for the carbohydrate. In the liver, glycogen, also in the muscle. But in the muscle, very fast source, very fast to hydrolyze. In the liver, a little bit slow. Yeah, and this will be stored. This type of the glycogen or carbohydrate will be stored as hydrolyzed granules inside the liver. The second point, very important, the contents of the glycogen inside the muscle and the liver. You have to know, despite of the amount of the glycogen in the muscle is greater than the liver, but the percentage is opposite because the mass of the muscle is greater than the liver. And we've got we explained example. In addition to that, we explained the one disease and we gave the symptoms of disease. This disease, Jarrett's disease, because of the deficiency of the glucose 6-phosphatase. Therefore, we don't have enough amount of the glucose, and that means lead to hypoglycemia, and the body will make substitution for the glucose by the ketone bodies or adipose tissue. Very important things here. You have to know to make depletion for the glucose from the liver need 18 hours fasting, then 1 hours vigorous exercise. By this way, you will make around zero level for the carbohydrate inside the liver and the muscle. By this way, the human body will reduce the weight by using another compound like triglyceride in adipose tissue. And we have started the first step, and we explained this two types of the enzyme, one of them hexokinase and the other glucokinase, to phosphorylate the glucose to glucose 6-phosphate. Okay, I'll give you 10 minutes. Don't be hesitated if you got any question. And thank you for your listening. Goodbye.